Hello viewers, today in this video we are going to see about Polytene chromosomes. Before entering into this topic, we must know what is called special chromosomes. Special chromosomes are chromosomes that show structural and functional modifications to meet specific cellular or developmental requirements. They are usually larger than normal chromosomes and are involved in intense gene expression. Polytene chromosomes or giant chromosomes, these are highly enlarged chromosomes that arise as a result of repeated rounds of DNA replication without subsequent mitosis or cytokinesis, and these process known as endoreduplication or endomitosis. This phenomenon leads to the formation of numerous sister chromatids that remain tightly aligned side by side, producing a single massive chromosome which can visible under a light microscope. With each successive round of replication, the number of chromatid doubles, resulting in a chromosome composed of hundreds to thousands of identical DNA strands bundled together. Polytene chromosomes were first described by Balbiani in 1881 during studies on the salivary gland cells of diphtheria insects. Occurrence and cell types of Polytene chromosomes. They are most prominently observed in the salivary glands of Drosophila larvae as well as in other metabolically active tissues such as the Malpighian tubules and midgut epithelial cells. The primary biological significance of Polytene chromosomes lies in their ability to increase gene dosage, thereby enabling extremely high levels of gene expression required for rapid growth and intense protein synthesis during larval development. Their large size and distinct structural organization make them an important model for studying chromosome structure, gene activity, and regulation. Coming to the structure, Polytene chromosomes show highly distinctive and orderly structural organization characterized by alternating dark bands and light interbands along their length. The dark band consists of highly condensed chromatin, which is relatively inactive in transcription, whereas the light interbands represent less condensed euchromatic regions that are actively involved in gene transcription. This regular alteration creates a precise and reproducible banding pattern that is unique to each chromosome and remains constant from cell to cell within the species. Chromosomal puffs or localized swellings that appear in specific bands of Polytene chromosomes at particular developmental stages or in response to environmental and hormonal stimuli. These puffs represent regions of intense gene activity where the chromatin undergoes decondensation to allow access to the transcriptional machinery. As a result, large numbers of RNA polymerase molecules and transcription factors are recruited to these regions, leading to active RNA synthesis. The formation of puffs is a dynamic and reversible process reflecting changes in gene expression according to cellular requirements. Exceptionally, large and prominent puffs known as Balbiani rings are involved in the synthesis of abundant secretory and structural proteins, highlighting the role of Polytene chromosomes in supporting high levels of transcription and protein production. Puff formation in Polytene chromosome is a highly regulated process that reflects precise control of gene expression. It is temporarily regulated, which means that specific puffs appear and disappear at defined stages of development, particularly during larval growth and metamorphosis. Puffling is also hormone controlled, with insect hormones such as esidisone playing a key role in activating or repressing specific genes by inducing characteristic puffing patterns. In addition, puff formation is responsive to environmental stimuli as external factors like heat shock can rapidly trigger the appearance of specific puffs associated with stress response genes. This tight regulation demonstrates a direct relationship between chromatin structure and gene activity, making Polytene chromosomes an excellent experimental system for studying gene regulation, hormonal control of transcription, and environmental effects on gene expression. And coming to the functions of Polytene chromosomes, increase gene dosage through repeated DNA replication, enabling high levels of RNA and protein synthesis without cell division. They support rapid growth and metabolic activity, especially in insect larval tissues, and allow regulation of gene expression through chromosomal puffs. Thank you for listening and watching the video. Have a good day.